Hi, good morning, everyone, or afternoon or evening. So basically, most probably you guys have heard in my lectures, I've basically said that I've decided to record my lectures, but this is not going to be recording exactly what I'm saying in the classes, but this is just going to be me going in a very much more slower pace, discussing concepts into more depth, and try to use more examples, the more that I can to make things more relatable for you. So use this more as a kind of a study guide more than anything else. Or if something you don't understand in the class or something and I'm not there to explain, use this video to help you. So in chapter 4, we will be speaking about the cell structure. So if you guys start to see, the first chapter till now, in the first chapter we really were talking about mostly, science and what biology is and how do we conduct, research in science, specifically in biology and all that stuff. And then we basically learn those things and then we start to apply them, on the smallest little particles, which we know as elements. Then, we know it's all these elements, combining together to create molecules, creating macromolecules. And then now, basically, we're going to be studying what these molecules will do and basically, how to create these organelles and how basically, these organelles are, let's say, organized within a cell. So let's start off with, a cell. So what is a cell? Cell, if you want to basically, define a cell, it's actually the smallest subunit of life. You know, all life contains cells, and cells is the smallest unit of life. And basically, an organism can be composed of one cell or multicellular, as we know them as prokaryotes, meaning unicellular, and eukaryotes, meaning multicellular. So the thing about cells, these guys are tiny, right? There are these small little objects that we cannot see, in the naked eye. We need a microscope to do so. And this, really goes back to, back in the day when we didn't have any microscopes. There was a lot of theory behind these cells. We didn't know much what they were. It's only until actually microscopes were actually invented we were able to actually discover cells. And this all, if you want to really track the science behind it, it all started off with Ibn Haytham, which is known as the first scientist. Most probably he's not the first scientist, but this guy was smart enough to record the scientific kind of methodology. So that's why he's regarded as the first scientist. So Ibn Haytham wrote the first book, which is known called the Book of Optics, Kitab al-Manazir, and basically, there he's really started to talk and really laid the foundations of modern physical optics. So he was really trying to understand, what light is and how vision is. Because if you think about vision, vision is all about light. It's basically these wavelengths of light that our eyes, have certain receptors that are able, let's say, to process these wavelengths and give us images. That's what basically vision is, actually. So he really laid out the foundations of it. Then later in 1585, so sorry, in 1632 or something around that time, with Zacharias Janssen, he came out with his father, and they basically used Ibn Haytham's book to create the first microscope. They claim it's the first microscope. Still debated whether yes or not. That's more into the history, which we really don't. We're not going to go into details about. However, these guys created the first microscope here, as you can see clearly, right here, where you have the eyepiece over here, and then you have the objective over here. And what these microscopes did, of course, they kind of made small structures look bigger through lenses. And this is all because of the foundations of Ibn Haytham. So this helped actually later on Robert Hooke, to use the inventions from Zacharias Janssen and his father to actually then, look into cells. So he really started to get, of course, these kind of plants and all these different specimens. And then he started to look like, these small little things, he called them cellulae, because for him, what cellulae actually means, little rooms. And as if you can clearly see, they actually look like small little rooms. I think there was this other scientist, Dutch scientist, which his name is quite long. Let me show you here in in your textbook. This Dutch Anton van Leeuwenhoek, and this guy basically had called them something else. He called it animalcules because he, as you know, after little animals. So that was the first descriptions of actually cells, little animals and little or these small rooms, these small little compartments. And these basically started off with the discovery of cells actually. But it was only until like later on, specifically these two scientists, Matthias Schleiden and Theodor Schwann, that they started to actually use microscopes much more and really start recording much more characteristics of cells. Specifically, Matthias Schleiden, what he did is he started to look much more in plants. And he started to realize that, oh, that these plants are just organisms that are composed of multiple cells put together. So, in his words, what he saw or what he said basically, plants are aggregates of fully individualized, independent, separate beings, namely the cells themselves. Meaning that, plants are just a bunch of cells stuck together. Theodor Schwann, 1 year later, said something similar, but this time he said it with animals. This guy was more into the animal, kind of basically studying more animals rather than plants, and he started to look into the tissues of these animals, and he saw that, like, damn, these tissues also composed of multiple types of cells. And because of these guys, Schleiden and Schwann, they're like, these came out, they started to propose the cell theory, which states the following. All organisms are composed of cells. Any living organism has a cell. So let's take an example, for example, like viruses. Viruses are not a cell, right? They do not have any cell. They're not alive. Only when they get it inside the cell do they become kind of alive. That's a bit more complex, but what I'm trying to simply say is that if it doesn't have a cell, it's not an organism. It's not living. So viruses are technically not organisms. They're dead. They're like semi. We call them semi dead because they need the cell to live. So always think about that. Two, cells are the smallest living things. So when someone tells you, for example, like we were talking about macromolecules or polymers, polymers are big structures that made up of monomers. Well, we are, let's say, and if you put that analogy with us, we are polymers, which are made basically of cells. The smallest monomer that we have is basically cells. That's the smallest living thing. And cells, the third one, cells arise only from pre-existing cells, meaning that cells do not randomly, combust or spontaneously just come out. No, they basically come from divisions of other cells, so either mitosis or meiosis. So all cells today represent a continuous line of descent from the first living cells, meaning that our cells right now come from cells that were dividing hundreds of billions of years ago. That's what I mean. So that that our cells right now didn't just spontaneously come out. That doesn't exist. They came from a previous cell back in the day. So one thing that we all know is that cells are quite relatively small. So have you guys never thought about that? Like, why are cells small? Why aren't they like much bigger? Well, it's an actually, if you think about it, it's an evolutionary adaptation and an advantage that cells are very small. Because if you think about cells, these cells are small little, let's say, compartments or tiny rooms where all the processes of life actually start to occur. This is where we synthesize proteins, this is where we synthesize carbohydrates, lipids, and DNA and RNA and all these different things that gives us life. So in these small little rooms, all these things are happening. But of course, as we started to learn, macromolecules or these polymers are made from monomers. So we need monomers to create these big polymers, to sustain life. So one thing that cells need to always have is this, import of stuff from outside. But of course, think about this also like when you're cooking. You basically import all the starting ingredients, right? You have all this stuff, you cook, and after you're done, you have a lot of waste. Well, same thing in the cell. When they start cooking their polymers and making all these polymers, there's a lot of waste that comes out. This waste, if we leave waste basically in the room, it's just gonna pile up, pile up, pile up until that room is basically uninhabitable. Well, same thing with cell. Cell starts to create a lot of waste that is basically going to become uninhabitable. You're not gonna. It's basically the cell is not going to be able to survive. So there has to be this import of stuff and export of stuff, which we like to call as diffusion of substances in and out of cells. So things have to always come in. So you have these kind of monomers to go inside, these building blocks that we really need to sustain life, and then the waste that we have, we need to produce it out. So we this all relies on diffusion of substances. So the rate of this diffusion, so basically how much import and export is basically going out, is affected by four main things: the surface area available. So this basically in a cell is the lipid membrane, because the lipid membrane is basically what's, the whole structure of the cell, is basically the whole outside of the cell. How big that basically cell membrane is, or how big the surface area of that membrane is, dictates how much stuff goes in and out. Because think about it, if it's very small, very little things can go inside. It's just because size availability, right? So that's one thing that can affect the rate of diffusion. Second is temperature. When you increase the temperature, you're giving a lot of energy to these molecules to go in. And if you lower the temperature, you really start to see stuff. One thing about proteins in general is that when you put in temperatures very low, proteins don't like it that. You're kind of not letting them, you're not giving them energy. So they're going to stay basically to preserve their energy. They're going to just, stay where they are. And then you have concentration gradient. So this is basically how much stuff you have in one place. So for example, in one cell, you have a lot of glucose, right? You don't need more glucose. So this concentration gradient is gonna tell the outside is like, hey, we don't need more glucose. Stop it. So that's one thing that can be affected. Distance. This is distance between a compartment inside the cell, so a place from a cell to the outside. So this could be the center of the cell, like the nucleus, to the outside. So of course, the bigger the distance, the more the affection of diffusion, how much you can affect this diffusion. And one concept that I want to really go into more depth is surface area availability. So cells are spheres, right? These really nice spherical objects. One thing about these spheres is, of course, you can measure spheres by surface area and by volume. Surface areas you can basically take as more like the two-dimensional size of it. So you're just looking just at, how big this ball is. The volume is you're taking also a more of a 3D plane, because, of course, the sphere is a 3D object. So now you're basically taking into consideration the whole space inside of it. So that's what volume is. And if you really want to calculate surface area and volume, surface area is this basically equation, while volume is this one here or over here. So let's imagine you basically have a cell that is 1 unit. If you calculate it, you basically will have 12.57 units squared. That's the surface area. And then volume will be 4.189 units cubed. So in general, there's much more surface area to volume. So if you put this, in a ratio, it's going to be 3, meaning that, there's much more surface area to volume. And for cells that are very small, this is what they have. They have a much higher surface area to volume ratio. And this is extremely important. Because think about this. As the volume increases, as a cell gets bigger and bigger, the volume is going to go, it's going to increase, right? And that means you need much more macromolecules, because now you need to sustain everything inside, because it's a much bigger um, cell. So think about this: a baby, needs this certain amount of food. When they grow older, the amount of food will increase because they're bigger. They need much more stuff to sustain. Well, same thing with a cell. If the cell is small, it needs less amount. And that's very important because the surface area is much then bigger. So this amount of stuff that you can take in and then export, the waste, is proportional then to, or basically higher than actually how much you need. So, how much the demand of what you actually have or need to live for that cell is actually, low compared to how much you can actually bring in. But imagine that this cell now basically goes from 1 unit to 10 units. So basically increases by 10. Surface area now becomes 1,257. So this actually increased by 100. But the volume went from 4.189 to 4,189. This increased by 1,000. So compared to surface area to volume, it increased 10 times. So if you look at now the surface to area volume, now suddenly it's completely decreased. There is much more volume compared to surface area. And this is a problem, because now you have a cell that needs a lot of building blocks and actually needs to also release a lot of more waste, and you don't have this big surface area to do so. And this is a problem. This is why cells are small. They cannot sustain such a big amount, such a, it's a gold, a big amount of building blocks while having a very small surface area. So that's what they mean here with the cell surface area. That the biggest characteristics of cells for them being small is that they have a bigger surface area compared to the volume. So they have a better, what's it called, diffusion of basically things going in and out compared to how much the cell needs, so the volume. So that's an advantage, honestly. So one thing actually I want to also talk about here, is that yes, cells are small, but there are cells that exist within our body that are quite big. Pure example is neurons. If you ever actually have the chance to look at a neuron in the microscope, please do so. They're very beautiful. They're actually quite very long, very actually tall, like very long cells, very big cells compared to your normal average cell, actually. So this kind of defies a bit, that cells are small. But the nice adaptation with these neurons is that they're very skinny. So their volume actually is not, it's not like a cell, like a sphere that, now basically the more you increase the cell radius, suddenly the volume increases much higher compared to surface area. Now it's a bit more different, it's more skinnier, and these kind of lost when applied to it. So that's why it's able to adapt. Muscle cells, for example, are quite also very big. They're not skinny like neurons, they're much, much bigger actually. Their way to cope with this is they actually have multiple nucleuses that are distributed along, and this helps it actually produce much more proteins and more other stuff to keep with the demand. So that's how these small little tricks of these other cells that are not that small are able to overcome this big surface area to volume ratio problems. So one thing that what I was starting off with this specific lecture saying is that cells are tiny, right? And we never knew cells that really existed. We always, of course, hypothesized and theorized that they must exist, right? And that's what we thought. But honestly, microscopes were the one actually that helped us, discover them and go properly more into their characteristics. But let's talk more about what microscopes basically are and all these kind of things. So one thing to understand really microscopes, you need to understand one term that which is resolution. Resolution basically means the minimum distance two points can be apart and still be distinguished as two separate points. That's what it is. Because now this term resolution, we really hear this, especially with TVs or anything have to do with images or stuff like that. It was asked, okay, what's the resolution of this TV or basically of this camera? Well, it's basically what it basically means is that how much can this object actually separate two points? This the distance, the minimum distance that basically this object can separate between two points. Because if you think about it, when we see an object, we actually have put 2 points. One point on basically its center, and the other one where it ends. So we need to basically that's how we see an object. We need to see a subject is where it starts and where it ends. That's why the 2 points comes into place. Because if we're not able to see basically these 2 points, we're not able to see an object. So objects that actually have basically the distance there are small as 100 micrometers or smaller, we're not able to see it because our naked eye will not be able to actually see that. Basically, if these two distances are 100 micrometers or smaller, our eye doesn't cannot see now suddenly those two small points that I'm saying. We cannot see the start and the end, so we see it as just one, and that is why we see it blurry. We don't see where it starts, we don't see where it ends. That's basically why we're not able to see a cell, because cells are actually around 10 micrometers actually. So if you see going now to the scale, a human XR is basically yeah, it's around 10 micrometers. So that's why we're not able to actually to see cells because we're not able to actually distinguish the starting and the ending point, and because that's the resolution of our eye. So microscopes, what they do is they try to enhance this resolution. They try to further magnify, make these objects look bigger. So we can now see these starting points. And there's two types of them, two types of microscopes that we will talk about. There's the light microscopes and the electron microscope. So these microscopes, as they say, they use two different kind of, let's say, things to look at specimens. Light microscopes use light, and electron microscopes use electrons. So basically, in the light microscope, what you do is you use a beam of light, and this light will pass to the specimens, and basically, you use two lenses, and these lenses will magnify. So light microscopes are actually, they're made of two lenses. One lens is there basically just to magnify the specimen, and the second one is basically is also then, for example, the first lens, it kind of projects the image in a more magnified more magnified. The second lens will even also magnify it even more, but more importantly, it will focus it on the back of our eye, and that's the importance of the second actually kind of lens, because if you don't have that lens, you're not going to be able to kind of focus it and focus it straight into your eye, because that's very important actually. So those are basically light microscopes. You can also call them compound microscopes because they're basically made of two um, lenses. So these guys basically will resolve structures that are 200 nanometers apart. So basically, any object that is 200 nanometers and above, it will be able to see it, which is great because that allows us actually to see cells in a very much good and a really high resolution. However, the resolution is, of course, is very limited to light, because people start to say it's like, okay, so with these two lenses, you can only see 200 nanometers. But why can't you add more lenses? Why can't you just magnify it more? Well, that doesn't work that way because you're using light, and light has a certain light wavelength. So if anyone's interested, how vision works, it's all about wavelengths. So we basically light is shined on stuff that then gives back wavelengths, and this wavelength basically is what our eye will take and process into an image. The wavelength actually of light that basically is reflected back is 200 nanometers. So this is why that's why the resolution is just 200 nanometers, because that's the wavelength basically of light. You cannot, the more lenses you do, that doesn't make anything else. Basically, because the wavelengths will coincide together and will be 200 nanometers. That's why. So that's why we use electrons. Because electrons, this time actually has a much, much smaller wavelength of zero point 0.2 nanometers. So with electron microscopes, you really resolve structures down to zero point 0.2 nanometers. That is crazy, guys, if you think about it, because hydrogen atoms, which is a small little atom, has the size of 0.2, 0.1 nanometer. So we're almost able to actually see a hydrogen atom. That's how good these microscopes are. So different to light microscopes, electrons use a beam of electrons, and this beam of electrons is shot to your specimen. And of course, the wavelength that is basically shot back is about zero point 0.2 nanometers, and that's why we're able to actually see or resolve these structures. So with light microscopes, the most commonly used light microscope is called this bright field microscope, which is basically you're shining light through the specimen. And what you do is you get basically kind of these images. This image is, if you see, what do you call it, this, the corners around, they're not that sharp. And that's why bright field microscope is quite good just to look at cells. But if you really want to see the details of these cells, it's not the best, honestly. You can basically start playing a lot around basically with what light, what the type of light you use, and basically what light you want to just absorb. So an example here is dark field microscopes, and where what you do is basically you shine light, and light that is reflected back because of this the specimen will be only taken. Anything that passes is not taken. So as you can see, anything that is basically empty space is black, because we're not basically taking that wavelength. We only taking the ones that are basically hitting you know the specimen, which is here. Then, of course, what you can do is you can start shining light at different angles or at different, time points to basically get a much more, better, let's say, focus on the edges, on the contrast of the specimens. And this is quite cool, I have to say. So these are different types you see in light microscopy. So this is basically in light microscopy. As you can see here, these colors are just then, what you can further actually do to these actually specimens is you can actually color certain compartments, and you do this using basic fluorescence. So let me show you here. So this is for some microscopes. So basically, what you do here is you tag certain proteins in your basically in your cell that give a specific color. So a pure example here, this blue. This is called DAPI actually, and this is a very nice basically antibodies that bind to your nucleus. And of course, these basically antibodies have a fluorophore. So these are small little basically chemicals you add to your antibody that basically give this blue light. So what you do is you create antibodies against the nucleus. you attach these small little fluorophores or these small molecules that make them blue. And then what you do is when you have the cell, you add these antibodies. These antibodies will go inside the cell and bind to the nucleus, giving this blue. So now basically you can clearly actually see just whatever is blue means it's actually the nucleus. This is, for example, here is the cytoskeleton. You can use antibodies that will only bind to the cytoskeleton and be green. This is basically other proteins in the cytosol. So when you put them together, this is beautiful because now you can see this. Okay, this is one cell, this is the nucleus, this is the whole cytoskeleton, and then this is the protein that you're interested in, and it's basically red. And you start can see it's like, okay, you can really start becoming look into more where these proteins are, because clearly these proteins are not the nucleus because they're mostly outside, they're not inside the nucleus. So you can say these are cytosolic proteins. So this is the beautiful thing about fluorescent microscope. And this whole thing is called immunohistochemistry, meaning because it's immunohistochemistry. It's basically, immuno is coming from immune system. Your immune system is the one that creates these antibodies against these structures, either the nucleus, the cytoskeleton, or the protein. And you're using these antibodies, at the chemistry of basically putting them into, for example, putting them into the antibodies to give color. So I can, for example, um, basically create an antibody that will bind to the nucleus, and I can actually give it a color of yellow, of green, of violet. That depends, of course, on the fluorophore. So that's the beautiful thing about fluorescent microscopy. So now let's talk about more electron microscopes. Electron microscopes are, wow, they're beautiful. They're really beautiful machines, but highly, highly complicated, sophisticated pieces of engineering, I have to say. So there's two main types. There's the TEM, which is transmission electron microscope, and scanning electron microscope, SEM. So they're very similar, in where they actually use the very similar concepts of basically electrons, but they just differ in small things. So TEM, what you do here is basically you shine electrons through your specimen, and of course these electrons are gonna basically blast and go through the specimen. So this specimen has to be actually very, 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 very basically thin. So that's the thing about TEM, that your specimens have to be very thin. So once that is done, great, the electrons are going to pass through it, and they're going to basically, of course, some electrons that are passing through very dense areas will lose a lot of the energy because, of course, now they've bumped a lot and they lose a lot of energy. The loss of these energy is basically what then gives the image. So let's imagine this. Take an example here. This is the cell. Suddenly start seeing, places, for example, here, this is much more dense. This is more dense compared to here. This is the nucleus. There's not, a lot of white, so meaning that's where a lot of empty space. But you start getting these really nice structures. So you get this guy, oh, this is a really nice, for example, like, lipid membrane. So the lipid membrane around here. And a lot of these kind of, let's say, kind of folds around here. And then, of course, you get to see, okay, this is the mitochondria. You get to see basically, again, much more rough folds, and this is more, most probably what's called the rough ER. These are small little things, little dots here, are most probably free ribosomes. So you get this again, start to see all these really nice, actually, kind of characteristics and depth into the cell. So this is TEM. Scanning electron microscope, so SEM, is a bit different. So what you do here is that actually, for example, you get your specimen. So in this case, it's the B, and you coat it completely in gold. Quite expensive, actually. But you then completely cover it in gold, and then what you do is you actually then hit a beam of electrons, but this time not like the TEM where basically it's going through. Basically, you're just gonna scan this, and what's gonna happen is that because it's covered in gold, basically these electrons will bounce off or just brush off the kind of what do you call it, the B, and basically what you then measure is the brushing off of these electrons, and these give you very, very nice um, resolution about actually the structures. So this is, for example, the B. This is the beautiful images that you can actually get. For example, this is a picture of basically osteoclast. So these are cells that love to degrade bones, and you clearly see that's actually degrading a bone. Here is basically is a cell. So these are HeLa cells. These are actually, if I'm not mistaken, they're liver cells. And then you clearly have actually here these little tiny virus particles, which is HIV. And here you clearly start seeing, that they're basically binding to the cells, trying to actually go inside. Here's a red blood cell, a platelet, and a white blood cell. So this is what it does. It loves to basically, these STEM can be really used just to see the outside structures of specimens in a very beautiful manner. So this is the main differences of TEM. TEM is basically you have a very thin specimen and you're shooting a beam of electrons that pass through. STEM, you're covering gold and you're basically letting, you're hitting the electrons, and the electrons basically, because when they hit the gold, they're going to brush off. And what you do is you measure that brushing off. And that really gives you a lot, an image of how this structure looks like actually. So quite beautiful, I have to say. So let's put things in a more of a scale of cellular components, right? So this is basically, an adult human being. So where basically this is this our basically this scale of size. This is the human eye. We can see, up to this kind of more than 100 meters. I think it's around like 200 meters, and then or more. And then basically the maximum we can actually go is around basically around 200 micrometers. So anything, 100 micrometers or less, we cannot see. This includes, of course, human eggs, our own eggs, red blood cells, HIV viruses, ribosomes, proteins or amino acid structures. We're not able to see that. What we're able to see is actually, cells of a chicken egg, a frog cell, a frog egg cell, these things we can see. So we can actually check vertebrate egg cells. That's what we can see. But any human eggs or basically, let's say, normal cells in our body, we're not able to see that. That's where we need microscopes. And light microscopes are able to see from 100 nanometers to 200 nanometers, sorry, 1 millimeter to 200 nanometers. So these basically is what you can see. You can actually see the mitochondria and up to a human egg, basically. With electron microscopes, we can go down to the amino acid resolution. That's how cool these electron microscopes are. Okay, so now I spoke about microscopes, and these microscopes allowed us actually to study a lot, many different types of cells, from many different organisms and whatnot. And one thing that we started to see with cells, with every single cell that we started to observe, they share four main characteristics. First one, they all have a region where actually DNA is present, be it that region a nucleoid or a nucleus. Difference between a nucleoid and nucleus: nucleoid is just a dense region where you actually, which is mostly in the middle, where you actually have DNA. That's what a nucleoid is. It doesn't have any cell membrane. It's not enclosed with a cell membrane. That's a nucleus. Nucleus is again another dense region where the DNA is located, but that is actually covered in a membrane. So nucleoid again is just, somewhere in the center where there's, the actually DNA is densely packed, but it's not actually wrapped around a lipid membrane. Nucleus is differences. Cytoplasm. All cells contain a cytoplasm, and cytoplasm is basically the inside of the cell, which is made of this cytosol. Cytosol is a liquid that it has inside mixed basically with a matrix of organelles and also all these macromolecules, the proteins, the lipids, the glycerols, ions, all these things. So it's the mixture of organelles and macromolecules that we actually have inside the cell and the cytosol, that's the liquid, that give it this very semi-fluid kind of texture, very similar to jello. So if you open up actually a cell inside, it's going to look like jello because it's literally liquid mixed with organelles, ions, macromolecules, all these things that give it this very semi-solid kind of state. So they all have a cytoplasm. Three, they all have ribosomes. Ribosomes are structures that create proteins. And as I said, one thing that distinguishes a cell from a cell is the type of protein they have. Because protein is exactly what helps us carry out function. So we need ribosomes. Without ribosomes, cells cannot exist. So that's one thing they also have. Fourth, they all need to have a plasma membrane. So, this membrane that is encapsulating the cell interior. So it's just a membrane that's outside the cell that is basically, kind of protecting the inside of the cell from the outside. So that's one thing. So now let's talk about prokaryotic cells. So these are organisms that are made of one cell. And these are basically either bacteria or archaea. Most probably you've heard of bacteria quite a lot, but archaea might be new to some people. Archaea are very, very weird for organisms. They're very understudied because These guys are quite weird to actually culture in a lab. That's why we've not done a lot of experiments on them because they're really hard to culture actually on. So that's why we're not going to really go into depth about it. We're just going to talk about much more about bacteria. So let's talk about in generally prokaryotic cells. Prokaryotic cells, as I said, they lack a membrane-bound nucleus. So they have a nucleoid. They do not have nucleuses, they have nucleoids. So basically, DNA is present in the central region, which is called the nucleoid. That's the biggest difference there. The cell has a plasma membrane, but they also have, actually outside of it, something called a cell wall, which I will be speaking more about it. They contain ribosomes, that's basically the biggest things about prokaryotes, or basically, by that's what they mostly have. But let's take a look basically of a structure of a prokaryote, and let's take here an example of this is most probably E. coli from just purely a structure. So you basically first have a capsule. Not all prokaryotes have a capsule. Some have and some don't. And this capsule is just a bunch of carbohydrates, that's creating an outside, literally capsule, like an outside barrier that's protecting, the prokaryotic cell. Then you have a cell wall, which I'll be going much more in depth what it is. And then you have a plasma membrane. Inside, you basically have like a dense region, which is the nucleus, and then you basically have here ribosomes all over. So basically, this is all entrapped. And then here, basically, you have these kind of hair-like projections, which are called pili. And these guys are quite nice. They're quite a weird structure. People thought these are used for motion. Not really. These guys are actually more involved in genetic material. They love to exchange genetic material through these pili, and they also basically help a lot, basically, bacteria to attach to host tissues. So E. coli loves this. E. coli is able to attach, for example, to our kidneys or to our ovaries, or basically, to create these massive inflammations because of this pili. This pili loves to basically attach to the kidney because of these guys over here. And they have this big, massive structure called the flagella that is mostly related for actually motility. Not this pili, but this guy over here. So let's talk about the bacterial cell wall. So bacterial cell wall is this guy over here that's outside the plasma membrane. This cell wall is actually made of a peptidoglycan, and this basically means it's just a mixture of carbohydrates and also peptides, small, short peptides, hence peptidoglycan. So here's where you have the beta glucans, so our beta glucoses that our body cannot actually be able to penetrate, or sorry, to cut because we don't have the enzyme for recognizing beta glucose. So that's the bacterial cell wall. And the good thing about this is simply with bacterial cell walls is that we're quite unique. You only will find these guys in prokaryotes. You can also find them in cell walls, but they're very unique uh, to bacteria, these cell walls. And this gives us an advantage because if you think about it, if you ever go to work in any field that's related to creating therapies, could be pharmacology, could be drug research, could be in pharmaceutical companies, all these different kind of things. The first thing you will basically look into a drug is that okay, is it specific? So for example, in this case, you have bacteria that's colonizing the body. So can you create a drug that's going to specifically only attack bacteria and not harm normal cells? If you can, jackpot, you won. Because the most important thing is that you want to give a drug to a human being that will not, cause a lot of damage. That's just going to be very specific towards whatever is causing the disease. So in the case of bacteria, you want it just to go and kill bacteria and leave your normal cells normal. Basically, not to touch them. And this is the nice thing about certain antibiotics. Some antibiotics will only basically destroy or stop the synthesis of bacterial cell walls or just cell walls because our cells do not have a cell wall. So this is a unique thing that we can use to our advantage. So Archaea, the cell walls of Archaea are quite weird actually. They don't have a bacterial cell wall. They actually have phospholipid membrane. Their phospholipid membrane is not a biphospholipid membrane like in human cells, meaning it's actually a monolayer. But it's quite weird because if you remember in the macromolecules, phosphate, you have a one phosphate head and then you have basically two fatty acids that are binding to it. Well, the fatty acids combine to basically to two phosphates, one at the front and one at the back. So that's why it kind of gives you kind of very similar, structure to a phospholipid membrane, but it's not. It's actually made of just uh, one layer of phospholipids. So that's why it's also, very different. They're also inside that cell membrane. You have actually inorganic compounds that we don't have in our bodies, and that's what makes it quite weird to actually, culture these cells in a lab. You cannot just take them and grow them in a lab. They're quite really hard to do so because we don't understand a lot about the chemistry inside what's happening in their what do you call it, in their lipid membranes. So we're not able to actually properly grow them properly because of these actually properties. So one thing I want to talk about actually from prokaryotes, which is the last, is flagella. So remember, flagella are these big, kind of huge structures that give motility. Well, this is how they look like. So this is Vibrio cholerae. This is a very nasty, actually, bacteria. You find them actually a lot in oysters. You don't want to actually ever want to get this bacteria because it's actually quite painful. Quite painful. These guys are actually quite motile. They love to swim around. They're really professional divers, I would say. And what makes these guys very um, actually professional divers is this big flagella structures they have. So these flagella structures are, as you can see, this very long filaments, and this is how they look like, or they're composed of. They have these two rings, one that's actually in the plasma membrane of the bacteria, and the second one that's actually in the peptidoglycan cell wall. They're connected there, and then basically then you have a hook which is connecting to the filament. And what happens is that through basically hydrogens going inside, so these hydrogen protons, so basically this is a proton, this will basically cause this kind of ring to go clockwise, to start spinning clockwise, and basically because this is all connected to each other, this will then spin clockwise, and this will actually give or propel the Vibrio to go very fast. If you're ever interested to see that, just go on YouTube and put basically Vibrio cholerae motility. These guys are fast as hell. They're really, really fast. They're really quite cool to see. Nasty bacteria, but quite cool, and to see how they actually behave, I would say. So that's basically how the whole flagella actually works. So now let's go into eukaryotic cells. So these are cells, of course. The first things that come to your mind are animal cells and plant cells, right? These guys are a bit, well, not a bit, quite more complex than prokaryotic cells because inside um, inside there, everything is compartmentalized. So again, this is the word I'm trying to spell. It's just so freaking hard. I don't know why, but compartmentalization means are that things are basically organized in a specific manner. In prokaryotes, this doesn't exist. It does to a small degree in some bacteria, not all actually, but in eukaryotes, this exists in a high degree. So this is actually achieved by membrane-bound organelles, meaning that the organelles or these basically regions where the specific functions are carried out are basically compartmentalized by a membrane. So the membrane is actually what's compartmentalizing them into their own thing, but also into this whole system called an endomembrane system, which I'll be speaking about later. So that's the big thing about eukaryotes. Of course, nucleuses are membrane-bound, so that's why they have a nucleus, and they actually have a cytoskeleton that's helping supporting and maintaining the cellular structure, which we'll definitely go into much more details. So now let's see about animal and plant cells. Animal and plant cells are very similar to each other. They actually have very similar structures. They both have a cell plasma membrane, and they actually contain most of the same organelles. The only things that actually makes them quite different to each other is that plant cells have a cell wall very similar to prokaryotes, and they actually have chloroplasts and vacuoles. These are very specific to plant cells. So chloroplasts are these small little organelles that actually are used for photosynthesis. That's why we don't have them. We don't do photosynthesis. And vacuoles have a very specific, um, kind of let's say, functions that that help cells. So we'll go into more details about that. But let's have a look, about the general structure of an animal cell. So first of all, you have this big, massive structure in the middle. That is the nucleus. This is where all our basically DNA is stored. And then basically around the nucleus, you have these big sacs and basically tubes of membranes all around, like this basically the ER. This is the rough ER, this is the smooth ER, the Golgi, all these, and these are actually what are making the endomembrane system. This is a system that actually this because of this it actually helps to compartmentalize things, in different places and organize places actually. But you have all these kind of, let's say, membranes all around, and you have these all other organelles like the mitochondria, which is the powerhouse of this, basically the cell. You have peroxisomes, you have basically small little lysosomes, centrioles, and etc. All of these, we're basically going to speak about them and what they do. Now let's have a look at the plant cell. First of all, just have a look. Very similar, right? Small, of course, there are differences, but quite similar. Similar that they have a nucleus, they have all this endomembrane system that I'm talking about, the mitochondria, the peroxisomes, and etc. Of course, there are differences like the chloroplast over here, you have the cell wall, all different, which of course, different type of cells. So let's talk about the nucleus. The nucleus actually in eukaryotic cells is really, really quite very organized, very let's say specific structure. And what helped us actually understand the structure of the nucleus and then hence the function was mostly looking into actually structures of it. So a lot of pictures were taken using TEM and SEM actually, and these are the following. So the first thing they started to see with actually the nucleus is that this seems quite thick. So the membrane is quite thick, and then they realized like, damn, this is actually made of two phospholipid membranes, not one. The cell membrane of the cell is actually made of one, while the nucleus is actually made of two. So that's why it's a bit actually quite more thicker. It's quite interesting. Then they started to see these pictures, especially here where I'm pointing out with my cursor, these small little depressions or craters right here. They first thought it's like, okay, this is maybe just the structure, this is like, what it looks like. But no. Then they found out these are actually complexes that they call nuclear pores. So these nuclear pores are actually, are kind of literally a pore that is made of eight subunits of a protein that are basically, organized in this kind of eightfold symmetry. So there are eight of them that are connected and make basically a basket. And in this basket, there's these kind of, let's say, long little filaments around it, up and bottom. And people start to think like, why do these exist? Why would you want to have a basket inside a nucleus? Well, think about this. The nucleus, the biggest thing of the nucleus is that it has DNA. So one thing about DNA is that the first thing that it does is that it is transcribed into RNA. RNA needs to go outside to the cytoplasm to be then translated into a protein, right? So there has to be some sort of basically, of course, uh, kind of basically efflux of stuff or RNA molecules created in the nucleus to the outside, right? So that's what the pores does. It helps things go outside. But of course, things need to go inside because we need enzymes, other machinery to go inside, of course, to replicate DNA, but also to transcribe DNA, right? So there has to be a certain complex that is basically regulating what goes up, out, and basically and in. And these are basically your nuclear pores. So of course, this is not an exam like what I'm going to be saying right now. When I just said everything about the nucleus pore, and everything like that, yes, that will be an exam. But this is an extra information that we'll add just for interest. What actually happens, actually, which is really cool, you'll definitely learn this bit later, is that any protein or anything that wants to go inside, what they do is they attach into these filaments and they start to kind of crawl, or you kind of use it as a rope to basically go inside the pore. And once they go inside, they again attach again and basically make their way through it. Quite a cool and beautiful system, I have to say. So that's basically how it looks on the basically the outside. When they started looking more into the inside of it, they started to see this kind of meshwork of like these networks of fibers all connected to each other. Actually, to me, this actually looks like kind of this scaffold that you put when when you break your arm. They basically put this kind of, let's say, this kind of that network fibers all around your kind of arm, the kind of or like this kind of cast. That's what it looks like to me, like a nice cast to give basically a structure. This is what these are. These are called nuclear lamina, which are literally network of fibers that kind of help the kind of nucleus give its pure structure and kind of gives it like, kind of help it, keep its nice spherical structure. So that's what a nucleus looks like. So one thing about actually eukaryotes and prokaryotes that differ other than the nucleus is of course the DNA. The DNA in prokaryotes is circular. It's just made of circles. While in the one in the nucleus, not really. The one in the nucleus is actually quite linear, and that's what we like to call chromosomes. So in eukaryotes, the DNA is divided into multiple linear chromosomes. So basically, they're literally linear pieces of DNA. And remember when I was saying about DNA, that DNA, if I wanted to stretch DNA from here, let's say from here, from Beirut, I can easily go to Cyprus, and even to the end of Cyprus, not just the beginning of it. It is very long. So you need to actually then basically really fold DNA a lot to put it in such a small cell, right? So how is this done? Is by basically getting proteins that are gonna come and basically, bind to the DNA and push it and help it fold. So when chromosomes basically are in contact with proteins that are basically helping it fold, this is called chromatin. So when anyone tells you, chromosomes, first thing that comes to your head is just, oh, he's talking about the linear DNA. When someone says chromatin, it's, oh, he's talking about, oh yeah, the DNA and plus the proteins are helping it to fold. So that's the differences. So that's basically what are found in the nucleuses of eukaryotic cells, these kind of chromosomes. While in prokaryotes, it's more circular. So now let's talk about ribosomes. As I said at the beginning of this lecture, it was that ribosomes are found everywhere because they're found in Archaea, bacteria, eukaryotes, so basically both prokaryotes and eukaryotes. They're found everywhere because these guys, their main function is just to create proteins. So we need to create proteins. If a cell wants to be alive, it has to be actually, it has to create proteins. And if you think about it, actually, there is a chemical reaction that we call fixation. And this is basically when you paralyze a cell. It's basically you kind of make a cell into a zombie, meaning that it's not alive, and it's not dead. It's literally in this quasi kind of sense, and that's called fixation. And in fixation, it's simply you add certain substances or compounds that what they do is they stop protein translation. Once you stop protein translation, that cell is basically kind of paused. It's just going to stay there and nothing's going to happen. And those are very good for us because sometimes what we like to do is get specimens, put this fixation, and they stop it at a specific moment, and then we can see what happened at that moment. So it allows us, to do all these different type of things to it, like add certain antibodies, which takes a lot of time, and we're still able to catch that moment. So that's the view. That's how important protein is. So going back to ribosomes. Ribosomes, of course, are found everywhere, and ribosomes basically are a complex of proteins and also RNA. So basically, RNA and which is called ribosomal RNA and protein come together to create a ribosome. And these then complexes basically take messenger RNA. So this is the RNA that is basically transcribed from DNA. It's gonna basically then attach to here ribosomal RNA, and then with the help of tRNAs, you're gonna get all of you're gonna get basically proteins. So translation starts to occur. So the details of all this process, and most probably you guys have done it in high school, but if not, we will be going them. If not in this lecture, definitely you'll get this a bit later. But ribosomes can be either free in the cytoplasm or in internal membranes. And that all depends, of course, on where the proteins are destined. If these proteins need to go basically to lysosome, if they need to go to the Golgi or the ER, they will be basically, these ribosomes will be in the endoplasmic reticulum. But if they're basically, these proteins are destined to stay in the cytosol, they're most probably going to be done in the ribosomes that are actually in the cytoplasm. So let's look at the structure about ribosomes. Ribosomes are actually, by itself, the proteins are made of two units, a small subunit and a large subunit. And usually these subunits are quite free. Only when mRNA actually comes out of the nucleus and now basically it's trying to be translated into a protein, these subunits come together, they join, and then of course with rRNA, with tRNAs that are coming also together, you start to create this synthesis. So that's what a ribosome looks like actually. So now let's talk about this endomembrane system. This is the system that I was talking about. Is this big, massive tubular system that are basically these kind of tubes and basically flattened sacs of membranes that are basically actually helping the cell compartmentalize. And this system is very important actually in creating and not creating proteins, sorry, but it's more in actually packaging proteins, in modifying them and transporting them wherever they need to be. That's what the endomembrane system is, and this is one of the biggest and most fundamental distinctions between a eukaryote and a prokaryote. If you actually think about it, prokaryotes do not have this endomembrane system, and this is actually a shame because proteins like insulin, for example, take a look at insulin. Insulin is very glycosylated, meaning it actually we add a lot of carbohydrates. We actually fold it a lot into its structures and everything. These have to be actually, produced in animal cells. They cannot be produced in bacteria because bacteria do not have this endomembrane system. So when you produce insulin, insulin will not be folded properly. It will not have actually the specific modifications that we needed. So it's actually not going to work because it doesn't have the proper structure. There's no function. So remember again, structure is function. So that's a big thing because eukaryotes are very expensive to culture. Bacteria, super cheap. Super cheap because again, bacteria they will just love to multiply. I can have hundreds of liters. I can do it. But having hundreds of liters of eukaryotes, you need very highly sophisticated systems to do so, maintain a lot of different parameters, and the food is much more expensive to prokaryotes, so bacteria specific. So that's one shame of it. That's why most of the proteins, that we give humans are done in eukaryotes because of this endomembrane system. And we'll talk about more in detail what this endomembrane system is. So the first thing in the endomembrane system is the endoplasmic reticulum. So when we actually started to look at the endoplasmic reticulum, which is mostly connected to your nucleus here, what you see is this again network of membranes that are basically they have this first of all this kind of flattened sacs actually and these tubes. And if you look at it basically here, this is a I think this is the TEM. Yeah, this is definitely TEM. This is the TEM picture. And again, you see again this little sacs and then these tubes around. This is basically, the endoplasmic reticulum. But one thing we started to see that this is a bit rough, like, well, this is a bit more smooth. So they gave it actually, there's like, okay, well, this is the endoplasmic reticulum, which can be divided into the rough endoplasmic reticulum and then the smooth endoplasmic reticulum. The rough endoplasmic reticulum has a lot of ribosomes attached to it. And this is what actually gives this kind of, let's say, grainy, very rough appearance of the rough endoplasmic reticulum. So because it has all of these basically ribosomes, the biggest function of this organelle is to actually produce proteins and then to secrete them. So basically, one thing is what they do is once they basically create these proteins, they secrete them inside here with to this lumen. So this is basically the inside. And the inside of the folds are either called cisternae or lumens. So remember, the inside of basically here is called cisternae or the lumen. So what happens is that these ribosomes are attached, they create the protein, and they push it inside. The protein then goes inside. It goes into the lobby, and it's waiting instructions. It's gonna be like, okay, you're this protein A, you should be in the and basically in the plasma membrane. So basically, it's gonna bring some taxis, which we'll be talking a bit later, and we'll transport it to its destination. Okay, you're this protein B, you should be actually no, you shouldn't be in the plasma membrane. You should be in the Golgi. I'll arrange a specific taxi to go there. So that's what's its biggest function is of the rough endoplasmic reticulum. The smooth endoplasmic reticulum is a bit different. It has very little ribosomes done. The biggest things that people started to see in the smooth endoplasmic reticulum is that it actually does a lot of synthesis of lipids and carbohydrates, but more importantly, it also actually serves a lot in calcium storage and also can serve as detoxifying agent. So it actually helps us detoxify, crap that we don't want in our blood, let's say, broadly speaking. So that's what was more basically, you know, the more function of smooth and the particulate. And because these guys have very different functions, the ratio between either you have more rough or smooth depends on cell function. For example, liver cells. Liver cells they need a lot. They need a lot of smooth endoplasmic reticulum because our biggest function of our liver is to detoxify. So it needs much more smooth endoplasmic reticulum. But for example, um, B cells. So these are immune cells that produce a lot of antibodies. Antibodies are basically, they're proteins. So these B cells, they need to do is produce massive amounts of proteins. So they have a much bigger ratio of rough endoplasmic reticulum. So, sometimes by looking how much there is, you can start to we see the cell functions actually. So now let's talk about the Golgi apparatus. So Golgi apparatus very similar in structure to the basically the plasma reticulum. They're these basically flattened sacs of interconnected membranes. So as you can clearly see here, and this actually has a certain phase. They have a cis phase and a trans phase because this basically will tell you how other transporting vesicles. These are basically the taxis that are delivering the protein from the ER that I was talking about. Uh, and basically, if they're entering the Golgi, that's called the cis phase. If they're leaving the Golgi, that's called the trans phase. So the biggest actually function of Golgi is to receive proteins or basically lipids or carbohydrates actually from the smooth or endoplasmic reticulum. And what they have to do is basically, properly package them. So in the case of proteins, it actually helps them to properly fold into its proper structure, modify it if it needs, a bit of carbohydrates there and needs some stuff. That's what it does. It also does that also for lipids and other, and basically, and also carbohydrates. So that's basically what a Golgi does. It helps further process and package basically, the incoming proteins or lipids coming from either the rough or smooth endoplasmic reticulum. So if they're coming, they come to the cis phase and then they go through the trans phase. So let's take here an example, of a protein that's destined to go outside. There are proteins that they need to be released outside of the cell. So, for example, the first thing that happens in the nucleus, DNA is transcribed. The mRNA goes basically here, goes outside to the nuclear pores, and then basically they will go to the ribosomes found basically in your rough endoplasmic reticulum. They'll be transcribed. They'll go inside basically of these folds, which again are called cisternae or lumens. And inside the lumens, they're basically awaiting instructions. They're like, okay, you need to you need to be excreted outside. Let's arrange a taxi. They get these bare vesicles, which are basically these literally small little, basically, vesicles coming from the Golgi. It's literally the proteins attached near here, the membrane, and basically these membranes start fuse out into these little vesicles. They're quite beautiful actually, and to see them actually in the microscope, these small little vesicles. So then they basically, as the taxi does, it goes, it transports them, and then basically they will fuse with the Golgi membrane, because the membranes of the Golgi and the endoplasmic reticulum are quite, basically, they're phospholipids. They're quite similar. So they're able to fuse, and then basically once they fuse, they release the cargo inside. Now, basically the protein is in the Golgi, and there it's going to start, properly be processed. It's gonna be, um, folded, modified, all these things we're talking about. And once that's done, they basically will, be transported outside. So again, the Golgi membrane, sorry, the Golgi will arrange a taxi, so which is its own lipid membrane that's going to be projected outside through the trans phase and basically be released outside. Because then, of course, this phospholipid membrane is basically then able to fuse with the cell membrane because they're made up together and they fuse really beautifully. And when they fuse, they release the things outside. So that's how basically this endomembrane system works. And this thing does not exist in bacteria. Remember that. So now we have other basically things that are part of the endomembrane system, which are called lysosomes. Lysosomes are these actually very small little secretory vesicles that we're talking about, but these guys contain very specific proteins. These proteins are quite vicious, meaning that they love to break stuff. That's what they do. They just love to break big polymers. So having these proteins just outside in the cytosol can be very dangerous. You just cannot do that. You're going to create a massive mess, right? So best thing to do them is actually keep them inside a membrane-bound digestive vesicle. So once they're produced, these proteins here, just keep them inside. So these lysosomes are kept inside and they're kept there because they have a very important function. Two very important functions. They recycle all organelles and they digest foreign matter, which is basically through phagocytosis. We'll talk about that right now. So organelles, like for example the mitochondria, all of them basically need to be renewed because they get old, and you know what? If they become old, they can start doing mistakes, and mistakes in cells gives us basically really a lot of diseases if you think about it. So we need to always make sure that all the machinery or all our organelles are in tip-top shape. So we need to always recycle them. So one way of recycling them is the lysosome comes. For example, here this is old or damaged organelle. This is the mitochondria. Is gonna fuse with this whole mitochondria, and basically now basically the mitochondria will be exposed to basically all of these basically kind of proteins that are breaking down macromolecules, which is basically what this whole thing is made of. It's all big bunch of macromolecules creating a basically an organelle. So the lysosome basically, and the proteins inside the enzymes will just gonna break it completely back to its single blocks. So then it can be recycled back to do new organelles. That's one basically of the main functions of lysosomes. Second one thing that cells like to do is called phagocytosis. Phago actually means eating. Cytosis means cyto is cell, is of. So eating of the cell. That's what it means. So cells like to do is they like to basically take up things from outside and basically, for example, big macromolecules, or they can be actually other stuff that we need to be broken down. So they'll take it up into again to this food vesicle, and they'll basically fuse with the lysosome. So now basically the food inside this vesicle will be exposed to all of these enzymes, and they'll degrade it and basically help with digestion. So those are basically lysosomes. There are other smaller, also very similar to lysosomes, basically other smaller, basically let's say vesicles that have different enzymes, and one of them is this peroxisomes. Peroxisomes have a different types of enzymes that are more related to oxidation. They have enzymes they love to oxidize. And oxidization means is basically giving oxygen. That's what it basically means. And in basically where you're doing, let's say, the polymerization of polymers and all that stuff, that can be very lethal. So you also again you cannot have these enzymes just fleeing around. So you again have them very similar to lysosomes. You have these basically oxidative enzymes encapsulated in a membrane. So that's what peroxisomes are. They are basically these ones that have oxidative enzymes, and they love to do what they got their name because what they do is they get water and give oxygen, and when you give water oxygen, suddenly it goes from H2O to H2O2. That's hydrogen peroxidase, and that is very, very, very dangerous. That H2O2 is very actually can chemically can create a lot of damage. So you cannot have that there. So that's why it will take it, it will create it, and they will break it inside because these peroxisomes actually have the enzymes to break it and to this basically into these byproducts which are harmless basically. So this is basically the peroxisome structure. They're actually made up of this composed of mostly of proteins actually, and they're these really large kind of structures. So vacuoles. Remember, vacuoles are one of the things that we actually were talking about before that are only found in cell and what do you call it? In plant cells. They're not found in animal cells. They're only found in plant cells. And these guys, if you have a look at them, if you actually really do actually a microscopy, you're going to see basically almost kind of this empty space. And literally, that's what a vacuole means. It's basically a big empty space. But believe it or not, it has a lot of function. One of the biggest functions of vacuoles is that, of course, it stores a lot of pigments, a lot of ions, a lot of waste, a lot of basically water inside there. But it also has many different types of. There's many different types of vacuoles. There are basically the central vacuoles in plants, which can be used for storage, like the storage vacuoles. But some of them basically also can contract, And basically, once these contract, they can actually make these plants bigger. Some plants when they grow, they don't grow because they're getting bigger by itself. It's because the vacuole is actually contracting and getting much, much bigger. And how they regulate these guys is that they have something called these. So basically, they're made of this kind of these tonoplasts. So which are basically they're basically this is what it's made of. These tonoplasts, and this tonoplast actually has a lot of these water channels. So these are channels that let water go out and inside, and that's how they can get bigger and they're smaller. And basically, that's also tonicity tonicity is basically the measure of osmotic pressure gradient. So that's why they call the tonoplast because they're able to actually, control osmolarity. So osmolarity is basically, the concentration of certain of water and solids inside there. So basically, they really because of these tonoplasts they can go bigger, whatnot, and accept certain things. So that's what vacuoles mostly are. So I'm gonna stop here. I will adjust this will be part one. Part two of the video, you can find it in my next link. Thank you for listening.